Hey Team Bio, welcome to your screencast about the cell membrane. So we are moving on from the big four. We have studied carbohydrates, fats and lipids, and now proteins, specifically enzymes, pretty extensively. And now we're going to move into talking about cell structure and function. So we've talked about all the things that make up cells, the big four, and now we're going to put it all together to actually talk about cells themselves. So the first thing that we're going to talk about is what creates the cell. Well, it's the cell membrane. The cell membrane is made up of all of the big four. But primarily, the major component of the cell membrane is fats and lipids. But there's also a lot of protein in the cell membrane. There is some carbohydrates. And there are some nucleic acids that play a role in signaling across the cell membrane. But we're primarily going to focus on fats and lipids and a little bit on protein. But fats and lipids is the main one. Okay, the primary component of a cell membrane is called a phospholipid. This structure should look somewhat familiar to you, regardless of what class you were in last semester. Both Ms. Gartner and I talked about this structure, at least briefly. Okay, a phospholipid is composed of kind of two different regions. One region is the phosphate head. This region is very polar, and as a result, it's really hydrophilic. It loves water. The other region of the phospholipid is composed of two fatty acid tails. The two fatty acid tails, you'll notice one is a straight line. This is denoting that it's a saturated fat. So there are no double bonds in this fatty acid tail. And then the other fat is kinked. So this means that there's a double bond in this molecule, so it's an unsaturated fat. It's important to have a mixture of saturated and unsaturated fat in the membrane because it helps control the fluidity of the membrane. Remember that saturated fats are solid at room temperature and unsaturated fats are liquid at room temperature. If you have too much of one or the other, then your cells will be too stiff or they'll be too liquidy. So you need a mixture of both. Okay, these two fatty acid tails are very non-polar. So as a result, they're very hydrophobic. Okay, so the cell membrane is composed of hundreds of thousands of phospholipids. When you get a bunch of phospholipids together, and oh no, I'm sorry, I'm gonna move this. All of you folks who wrote in pen or pencil even, I apologize. Okay, when you take a phospholipid and you get a bunch of them together, they start to aggregate in a way that is very predictable. They form a bilayer. So usually this is happening in a watery solution. So there's going to be water on this side and also water on this side. So when you mix a bunch of phospholipids up together with water, they spontaneously form a bilayer. So a layer of two. And why do they spontaneously form a bilayer? What's the most energetically favorable conformation for these molecules? Because it puts all of the hydrophobic tails together. Away from the water, here and here, and it puts all the hydrophilic heads together facing the water. So it's the most energetically favorable position for all of these molecules. Now you might be asking, well, what about this region in here? What, doesn't water just creep in and start to interact with this side? Wouldn't that be unfavorable? And the answer would be yes. So cells don't just form a bilayer that's really tiny. They actually form a bilayer that's an entire three-dimensional ring, where this is a cell. It's composed of millions of phospholipids that form a ring. The ring is itself a bilayer. It has a hydrophobic interior and a hydrophilic exterior. There's going to be water. Oh, cells have two L's in them. There's going to be water that is outside the cell. And then water that's also inside the cell. And because there's essentially a layer of fat trapping this water layer in here, we can create an internal environment that's different from the exterior, because the hydrophobic layer prevents things from passing through. I don't know why the second L keeps deleting. A cell, two L's. Okay, so here's my drawing of it. Here's potentially a better drawing of it. This image right here is actually showing you kind of like a three-dimensional view of a cell, where we have a phospholipid bilayer, and you can see that a bilayer extends throughout the whole cell. We've got water on the inside of the cell and water on the outside of the cell, but because we have this hydrophobic layer, the water or the polar things can't pass through very easily. And so we can create an internal environment that's different from the external environment. So that's an important function of a cell membrane. So cell membranes are what we call semi-permeable. They only allow some things to pass through, which I guess is linked to this. Okay, So this image, my kind of lacking image, and then this image of a cell, they're both actually missing some of the complexity that a cell has. So the model of a cell membrane that we're going to be talking about is called the fluid mosaic model. Okay, let's first talk about the fluid part. Remember above I said that the cell membrane is composed of one saturated fat and one unsaturated fat, and that helps control the liquidity of the membrane. Well, every component in the cell membrane has free ability to move and diffuse around within the cell membrane. So this phospholipid might be over here when we start a timer. If we were to label it, like radioactively label it with a tag, and then we start a timer, we wait 5 minutes, and then we find out that 5 minutes later, this phospholipid is actually over here. It's moved within the cell membrane. So nothing within the cell membrane is fixed. Everything is moving around. And then we call it a mosaic because unlike this simplified cell here and here, they're not just phospholipids that make it up. They're also embedded proteins and glycosylated proteins, which means sugar proteins. That span the membrane and are embedded within the membrane. So here we have an example of 3 different kinds of proteins. Here's a protein channel. Within the membrane. Here is a glycosylated or a sugar protein embedded within the membrane. And then here is a receptor protein. That is going to bind a signaling molecule. And then activate an intracellular protein, which might break away and go off into the center of the cell. So this fluid mosaic model of the cell membrane shows us that nothing is fixed in the membrane. It's all freely diffusing around, and then it's not just phospholipids. It's a mosaic of a bunch of different components, primarily proteins. Okay, that's all.